Thank you, Geordie and Brinky, for the introduction. And I'd just like to say thank you to Zespri and Juliet in particular for inviting me to speak, to give me the opportunity, and to the Redette Institute for hosting as well. So we've heard a lot over the last few days about the health benefits of kiwifruit. They're real. There's laxation, glycemic impact, which we've just heard about, the immune benefits, and vitamin C earlier this morning. But I'm going to be talking about the prebiotic effects. And so Renee's introduced my talk quite well. But I just want to touch again on the definition. So a selectively fermented ingredient that results in specific changes in the composition and or activity of the gastrointestinal microbiota, thus conferring benefits upon host health. So it's the compounds in kiwifruit that can make it through to the colon and start to affect beneficial change. And I mean, we've had a bit of talk about EFSA and regulatory authorities like EFSA quite rightly judge what is termed as a prebiotic. And there's probably a handful of known prebiotics in the world, lactulose, inulin, fructooligosaccharides, and galactoligosaccharides. So I'm not saying that kiwifruit is a prebiotic. I don't think that's probably ever going to happen. But it can exhibit these prebiotic effects, and that's what we're going to be talking about today. So like I mentioned, there's 2 to 3% non-starch polysaccharides or fibre, and so those are the pectic polysaccharides, semicellulose, cellulose. And these can escape digestion by our relatively limited genetic potential of the human body compared to the bacterial complement, and so reaching the large bowel and having some effect there. So just one quick slide on the gut microbiota, because I know Renee was speaking before me, but it's been associated with many aspects of health. So these have been touched on before, local and disease-related, IBS, IBD, but also systemic effects, allergy, asthma, the gut-brain axis, and amongst others, obesity. So it's really the colon is this organ. The bacteria in the colon is almost like an organ within an organ, and we're just starting to understand how much of an effect those bacteria and their metabolism have on our health and well-being. So the short-chain fatty acids are probably quantitatively the most produced acids or metabolites in the large bowel. Branched-chain fatty acids as well, and then gases such as hydrogen, carbon dioxide, and methane. So there's the acetate, propionate, and butyrate are some of the 3 big ones, as well as lactate, formate, succinate, and branched-chain fatty acids like isovalerate and isobutyrate. So I'm going to really be focusing on these 2 publications that came out as a result of my PhD, which I finished last year. And so there's been quite a number of in vitro and in vivo studies on the effect of kiwifruit on the gut microbiota, much more probably on other aspects. But there's been one human trial, I believe, in Singapore a few years ago, but most of the work is focused on in vitro studies, and that's what my area is, and that's what I'm going to be talking about today. So I'll just briefly touch on this model that I used in both studies. It's one that was validated in the United Kingdom, the University of Reading. So one of my supervisors was Glenn Gibson, and he kind of started running these models years ago. And so basically the idea is this little vessel, which is a couple of 100 mils, mimics the large bowel microbial community. So that's why it's brown. And you can control several variables. So pH control is probably one of the more interesting ones and important ones. So there's a little probe that sits in, senses the pH in real time and adds in acid or base automatically as required to keep it between a certain pH range. And this mimics what happens in our bodies with carbonate production. Temperature is kept at 37 degrees. Oxygen levels are kept to a minimum by bubbling through nitrogen. And then we have to obviously add the bacteria, which is a fresh fecal sample. And then there's can be a bit of variation between individuals in their microbiota composition, which I'll touch on later. And so we do run it in triplicate and take an average of those results. And then because it's got the pH controlling component, we can run this for 24 hours or even 48 hours. And we do have other models that are a bit more complex that we can run for 6 weeks or so that mimic 3 stages of the colon, but I'm not going to talk about those today. So the first study was green and gold kiwifruit. It was Hayward and actually Hort 16A a few years ago. We used 16S ribosomal RNA gene sequencing or high throughput sequencing. So I think that's probably one of the advances in this study over some of the earlier ones which just use qPCR, which looks at several bacterial groups as opposed to this whole community effect. And then some metabolomics looking at gas chromatography and NMR spectroscopy to look at not just the short-chain fatty acids, but some of the other players and how they're changing. So there's a picture of some of the vessels running with the pH controllers. So straight into the results. Really, this is a relative abundance graph, quite busy. But basically, the green bar is around the green kiwifruit vessels, and the gold around the gold kiwifruit vessels. The light blue is what I use for my negative control throughout the presentation. You'll see that. And from the left-hand side, within each of these boxes, 0 hours, 5 hours, 10, 24, and 48 hours of fermentation. So as time moves on from left to right. And effectively, it's a percentage or a fraction of 1. So this sample at 5 hours, those are the percentages of different bacteria that make up that sample at that time. And so the 4 main phyla we see in a fecal sample are Proteobacteria, Firmicutes, Bacteroidetes, and Actinobacteria, and then different colours or different shades within those 4 main phyla are depicted by most of those families and genera of bacteria. So it's quite busy and hard to tell exactly what's going on, but basically some of the more interesting ones are the yellow, which is the Bacteroides species, genus, and the crimson, which is Bifidobacterium. And so I'll just zoom in on those in this slide. So for the light blue, for Bifidobacterium, you can see that it stays relatively constant over the course of the experiment. Gold kiwifruit didn't show an increase, but green kiwifruit, we start to see this quite big increase by 24 and 48 hours. So from less than 2% up to 6, almost 8%, so almost a 4-fold increase. Bacteroides, so these, again, the negative showed relatively constant over time, but both green and gold showed this increase from about 5% up to 15, almost 20, and then dropping back down again by 24 and 48 hours. So Bacteroides represent a relatively large group of bacteria. Some studies say that they can make up to perhaps 20% of the composition of the gut microbiota, so a very large genus with dozens and dozens of species. And so is it a good result? Well, yes, it is, because they've been shown to produce beneficial short-chain fatty acids, and propionate is one of the short-chain fatty acids that they produce in particular abundance. And they can also access some of the carbohydrates that are otherwise recalcitrant and hard to access, and other bacteria don't have the ability to do that. And there's been shown that there's a symbiosis between these 2 bacteria in different studies where the Bacteroides are able to unlock some nutrients and Bifidobacteria come in and eat them. And so the fact that we're seeing this increase earlier in Bacteroides and then a later increase in Bifidobacteria could be something along those lines, this cross-feeding network. So looking at the organic acids in those same experiments, so the light blue is the negative control. And so we can see that acetate is produced in the highest amount in all vessels. Propionate is produced in the second highest amount. And butyrate is a little bit less. It's still there, but relatively comparable amounts to the negative control. And so that's what I mean by the being probably slightly less of a butyrate effect. But I think on a micromole basis, the actual butyrate in there is having quite a punch, quite an effect. Propionate has been shown to be involved in inhibiting lipogenesis and involved in regulating cholesterol synthesis. And there has been studies shown that it can be involved in modulating satiety as well. So, and we do know that Bacteroides, some of the biggest short-chain fatty acids they produce, they're shunting towards the propionate pathway. Bifidobacteria in particular produces acetate, so we're kind of seeing consistent results between the abundance and the types of metabolites they're producing. The three on the bottom, lactate, succinate, and formate, are intermediate short-chain fatty acids. So you can see that most of them spike up after 5-10 hours and then drop down again by 24 hours and 48 hours because they're produced early on in their fermentation, and then they can be onwardly converted into other short-chain fatty acids such as acetate and propionate. So the second study, we used the SunGold ZZ002 kiwifruit. Again, 16S sequencing, but a more advanced method. So the first study used 454 sequencing. This used Illumina MySeq sequencing and gas chromatography and LCMS. So this heat plot shows just a different way of showing the bacteria and how they're changing over the course of the experiment. So everything below the yellow lines was in the gold vessels. The blue was the negative vessels. Here, might not be able to read, but basically that's time 0, time 5, 10, 24, 48, so from left to right over time. And then the top 22 bacteria that were changing over the course of the experiment are on the right. And those are within the main phyla, so the orange are Firmicutes, the purple is Bacteroidetes, blue is Actinobacteria, and red is Proteobacteria, and green is Verrucomicrobia, which has Akkermansia in it, I think someone mentioned yesterday. So initially we see this drop-off in some of the Firmicutes, which is more of a result of the change from the in vivo situation through to putting it into a test tube and trying to recreate the large bowel. But that starts to correct itself over time. But if we once did the statistical analysis on this, found that the Parabacteroides and Bacteroides and Bifidobacterium were the significant increased bacteria over the course of the experiment. So it's kind of backing up what we saw in the first study and adding in Parabacteroides as well. So this is a beta diversity unweighted biplot. So it's a way of looking at how different, taxonomically, how different samples relate to each other or their similarity or dissimilarity. So samples that are quite close together are probably quite close in their taxonomic composition bacteriologically. So like I mentioned before, we do these studies in triplicate because there's quite a difference between the microbiota of you two and myself, and so we need to try and account for those differences. So donor 1 in red, donor 2 in green, and donor 3 in purple. The lighter dots represent the earlier time points, so that's time 0 for purple and red and green, and towards the darker dots over time, and so we can see that over time it's moving this way and this way towards the Parabacteroides, Bacteroides, and Bifidobacterium, Arachnospiraceae as well. So this is quite a key finding because it shows that even though the starting point of these studies were quite different, so they had different bacteria, which we know that happens, kiwifruit was able to drive the direction of bacteriologically towards more commensal genera or beneficial bacteria. And the negative controls, I didn't put them on here, but they didn't show any migration at all. So this is the organic acid concentrations for that study. So the gold here on the left, acetate, propionate, and butyrate, just showing the big three, and then acetate, and propionate, and butyrate for the negative. And so you'd expect them to increase over the course of the experiment, which they do. But the ratio for this study was 67, 26, and 7 for acetate, propionate, and butyrate, respectively. So you normally see a 60-20-20 ratio or 3-1-1. So we're seeing a little bit less of the butyrate, but more acetate and definitely more propionate. So that kind of fits with the first study as well. And so this is some of the LCMS results showing a couple of the metabolites that we quantified in the vessels from kiwifruit. So quinic acid, citric acid, and malic acid are organic acids that are native to kiwifruit. They live and they exist in kiwifruit, and in relatively high amounts, so up to almost 2% for those three, and most of that 2% is probably quinic acid. So almost maybe over 1% of the weight of kiwifruit is quinic acid, which is quite a high amount, and that kind of sets it apart from other fruit, makes it quite unique. So I haven't mentioned so far, but of course, you know, the colon is first, anything that sees the colon has to first traverse through the mouth, stomach and small intestine. So like all the other studies, we have to perform a simulated digestion before we add anything into the colon models. And so the fact that we're seeing here at time 0 in the models, these quinic acid, citric acid, malic acid and other compounds, it means that, at least in the laboratory setting, they're able to survive simulated digestion and reach the colon intact. And so they could then perhaps have some effect on the microbial composition therein. So I haven't been able to prove that they're being metabolized by bacteria, because they're dropping off by 5 hours, 10 hours, and then they're gone by 24, 48 hours. And so one of the other options is that they could have just been degraded by the conditions. But the fact they've passed through the very acidic conditions of digestion and enzymatic conditions makes me think that perhaps they actually are metabolized by bacteria. And we know that they are substrates that bacteria can access. Quinic acid is involved in the regeneration of amino acids, aromatic amino acids, such as tryptophan, which is why I put that up there, because you can still see those levels of tryptophan at similar time points. That's through the shikimate pathway, and citric acid and malic acid in the citric acid cycle, and involved in the regeneration of ATP. And ecofoil 3-glucoside is just one of the polyphenols. It's a hydroxycinnamic acid, which we found to survive through digestion as well. So it's just showing that not just carbohydrates or fibre are making it through to the gut from kiwifruit. So really, in conclusion, just to say that despite the notable donor inoculum differences, kiwifruit was still able to drive the profile towards a more commensal genera, and the supplementation in vitro promoted the commensal Bacteroides, Parabacteroides and Bifidobacterium, and this resulted in a slightly higher proportion of propionate, and organic acids and polyphenols can survive simulated digestion and reach the colon, and thereby potentially acting as substrates for fermentation. So like any PhD project, it's perhaps thrown up more questions than it's answered. And so there's one of the probably most obvious ones that I ask myself after this work is what fractions of kiwifruit are responsible for causing those effects that we've just seen. I mean, we're pretty sure that most of those effects are by polysaccharides, but we haven't really characterised that, and didn't really have time to look into that in detail. But of course, these other organic acids and polyphenols may be having a role, and there's also fatty acids, which are actually relatively high in kiwifruit as well, linolenic acid, linoleic acid, and oleic acid, which potentially could be causing effect. I managed to detect them as well, but as to whether they're actually causing a change in bacterial profile, not sure. But there's actually going to be a PhD student start in conjunction with Zespri and our group within the next few months to start looking at some of these questions and picking up from where I left off. Yes, so laxative effects, we talked about those. I mean, we saw the trial results. They're very real. I just wanted to leave you with a parting message about that. So Carleen mentioned a little bit about it, and John, what I find it quite fascinating that we don't actually know exactly what is causing laxation in kiwifruit. So is it fibre, is it oxalate or the raphides, actinidin, organic acids? Who knows? I think obviously there's perhaps a combination effect between them, but I don't think anyone really definitively knows. Someone will probably stand up and prove me wrong now, but I just want to throw another one in there. What about the bacteria? I'm slightly biased because I'm a microbiologist, but I think they might have a role to play. So these three studies, well, these two studies and a kind of letter commentary on them, came out all within the last few years, the last year actually. And so they were finding that stool consistency was associated with the composition of the microbiota, so the Bristol stool scale, and then they took fecal samples, sequenced, and they found many associations with diversity, richness, as well as the absence or presence of specific bacterial groups with high or low levels of, or high or low on the Bristol stool scale. So, I mean, it's probably a secondary effect, because you could say that fibre is causing the changes in bacteria, which then cause some of these effects. So it's fibre as well as bacteria. Organic acids may affect the composition of bacteria, so they're all interrelated, all linked. But I think it's probably something that warrants a bit of further research and study. So yeah, that's it. Thank you very much again to Redette and to Zespri for the opportunity to come and present here. To everyone in our food, health and nutrition team in Palmerston North, my supervisors, Richard Geary, Juliet and Glenn Gibson, and happy to take any questions. Thanks.